So we are starting the conversation about metabolic pathways that generate ATP and all the energy story within the cell. And we basically going to focus on mechanisms, how we extract energy out of the nutrients. Okay. So the term that is used for all chemical reactions in the cell is metabolism. Okay. Metabolism implies all chemical reactions, not just ones that provide energy in substances. Okay. Now there are two main types of reactions that participate in the energy exchange, and not only energy exchange, just reactions that exist in the human body: catabolic and anabolic. Catabolic reactions, they break down. Maybe it's a large molecule, maybe it's not so large, such a large molecule, but they break down. Anabolic reactions use the energy of ATP to synthesize, to build larger molecules. So catabolic reactions, for the most part, provide energy. So there are three main steps in catabolism of nutrients. So the first step is digestion and hydrolysis of large biological molecules, such as proteins or polysaccharides, into smaller building blocks that can enter the bloodstream and be further degraded. The degradation process is the breakdown of the molecules that we produced in the previous step into 2 and 3 carbon intermediates. And then the final step is the oxidation of this 2 to 3 carbon intermediates in the process is called citric acid cycle and electron transport chain, so that they can release energy used for ATP synthesis. So what effectively it comes down to is, if you think about your waste that you produce, chemical waste. I'm not talking about like waste from digestion, human feces, a lot of it is still large stuff. But think about what you generate. You effectively generate carbon dioxide, which you exhale. You generate water, which you also partially exhale and sweat out. And you generate urea, which you excrete, urine. That's pretty much all metabolic waste. That's it. Okay, so we're going to figure out how it works. So in this scheme, you can see that proteins are broken down to amino acids in the first stage, which is digestion and hydrolysis. Polysaccharides are broken down into monosaccharides in a stage of digestion, and lipids are broken down to fatty acids and also glycerol. Amino acids, through the process of transamination, I'm going too much to the right, so transamination and deamination, in the process of degradation, basically, they generate either acetyl-CoA, coenzyme A, basically 2 to 3 to 4 carbon intermediates, which then broken down citric acid cycle and so on. Fatty acids and glycerol, okay, in the process of beta-oxidation, okay, are also converted to acetyl-CoA, and monosaccharides in the process of glycolysis, broken down to smaller chunks. And then finally citric acid generates intermediates, oxidizes those intermediates, and that oxidation provides energy for ATP synthesis in the oxidative phosphorylation. So our goal is to discuss how each of these steps work. For this we need to talk about general concepts of structure in the cell, structures in the cell. So every cell, and I mean it, every cell, has nucleus, cell membrane, cytoplasm, cytosol included. Cytoplasm is cytosol, the goo, plus all the cellular organelles, and ribosomes. Okay? Most human cells, not all of them, but most human cells, also have mitochondria, which indeed is a powerhouse of the cell. Mitochondria is the place where ATP is synthesized. Now, if you look at mitochondria closer, you will see that first of all, it has two membranes, outer and inner. There is a space between the membranes called intermembrane space, and within the bounds of the inner membrane is the mitochondrial matrix. Okay? So this is the list. We already discussed. Cell membrane is the boundary. It separates the cell from the environment. Cytoplasm is everything within the boundaries of cell membrane with all the organelles and stuff. Cytosol is just a part of the cytoplasm. Mitochondrion is responsible for ATP synthesis. Nucleus is where genetic information is stored, and the ribosome is protein synthesis. So this statement is wrong. Let me explain why. There's a term that energy is stored. Why this statement is wrong? Hear me out. When the worst way to save money is to put them in a jar, right? So when you store money, you bring them to the bank. Am I making sense? So in order to get this money, you have to go to the bank or withdraw the money, go to ATM, right? ATP isn't a storage. It's a currency. It's constantly synthesized and it's constantly used. It's not a storage of energy by any means. Cells store energy using different molecules. But ATP is the universal molecule that fuels pretty much every cellular process. Okay? ATP stands for adenosine triphosphate. As you already learned, adenosine is a nucleoside that consists of the base adenine, sugar ribose, and three phosphate residues, also known as alpha, beta, and gamma, and the bonds between the phosphate groups. The bonds between gamma and beta and alpha those bonds, when broken, release a lot of energy. This is why we call them high energy bonds. Okay? Does that make sense? So when the molecule of ATP is hydrolyzed, I'm going to put a little water in here, the adenosine diphosphate is formed, and inorganic phosphate. So Pi stands for inorganic phosphate. Okay? You can see that this reaction releases a pretty decent amount of energy per mole. And then adenosine diphosphate, ATP, can be further broken down into adenosine. So ATP can be broken down to adenosine monophosphate and inorganic phosphate, releasing the same amount of energy. So I'm going to try to avoid the statement that it's the energy storage molecule, and I'm going to use the term energy currency instead. So right here, take a look. When you eat something and the food gets digested, and then the building blocks are being degraded and the small bits are oxidized, ATP is produced. Okay, in this image, this representation of ATP, you can see the adenosine portion. And these are phosphates. And then this ATP, the energy, is used for some kind of anabolic reactions, like synthesizing proteins, something like that. Okay, broken down to ADP and phosphate, and the energy for restoring ATP comes from food again. Okay, so just like currency allows us to link our different activities, ATP links different reactions. Because think for a second, if you would have to directly channel energy that is contained within carbohydrates into some kind of anabolic process like protein synthesis, you would need an insane number of enzymes, facilitators. Think of it, are you familiar with the term barter in economics? So let me explain. My profession is a teacher. I need to buy bread. Imagine that I go to a bakery and I tell them, how about I'm going to teach you 20 minutes of anatomy and physiology for a loaf of bread? That's a stupid idea, right? Maybe they don't need my teaching. So we use currency to organize the economy. Same way ATP is used by the cell to organize and link all the chemical processes. So first, let's talk about digestion. So breakdown of large molecules to smaller ones. So first is the breakdown of carbohydrates. So carbohydrates are digested in two locations, in your mouth and in your small intestine. In your mouth, saliva contains the enzyme called salivary amylase. This salivary amylase breaks down the glycosidic bonds in starch, specifically amylose and amylopectin, and that results in smaller oligosaccharides such as maltose, polysaccharides dextrins, can be more than 2 glucose units, or even monosaccharide glucose. So let's give them the proper assignment. So disaccharide, two glucose units. Glucose is monosaccharide, and dextrins are oligosaccharides. So when you swallow those partially broken bits, they go into the stomach where no carbohydrate digestion occurs because it's too acidic. Eventually, those bits and pieces reach small intestine. In the small intestine, pH becomes slightly basic. Okay, the pancreatic amylase. I'm going to actually write down the name of the enzyme for you. Amylase breaks down the remaining dextrins, and the enzymes that produced in the cells of your intestinal mucosal lining. There are three enzymes that digest carbohydrates. It's maltase, breaking down maltose, sucrase, that breaks down sucrose, table sugar, and lactase, that breaks down milk, sugar, lactose. Okay? They break those down into monosaccharides. Monosaccharides are absorbed through the intestine in the wall and, into the bloodstream. And then, since you get fructose from sugar and galactose from lactose, milk sugar, they have to be converted to glucose in the liver. So the places where carbohydrates being digested are specifically mouth and small intestine, and another organ that must participate in that digestion is, I'm going to highlight it in a different color, it's a pancreas. My point is, pancreas isn't an empty organ, but it provides enzymes for the breakdown of smaller bits of saccharides in the small intestine. So this scheme actually quite nicely shows you what happens in each compartment. So in your mouth, polysaccharides are broken down by salivary amylase into dextrins, maltose, and glucose. And then in the small intestine, dextrins are broken down by pancreatic amylase into maltose, and since you probably eat some dairy or drink some milk, there's lactose involved, and if you add sugar to your tea or coffee, sucrose is involved. At the end of the day, those three are disaccharides, and they are being broken down to monosaccharides. Good? Everybody's with me? Okay, awesome. So some people are intolerant to lactose. Some people are intolerant to lactose genetically. They produce very low levels of the enzyme. In some people, the amount of the enzyme lactase that we produce goes down as we age. Right? Because milk is not the main component of our diet as we grow up, right? Because people still drink milk, but they also eat a bunch of other stuff. Okay? Now, it can be to a different degree, but basically what happens if people with lactose intolerance will eat lactose-containing products, they will have bloating, diarrhea, and so on and so forth. It can be helped by ingesting the drugs that contain lactase, just to help you digest lactose, or avoid products that may contain lactose. I think I'm going to go through digestion, and we're going to call it a day. So, fats. Are fats water soluble or not? They're not. Everybody agrees with me? Yeah? So, your intestinal environment is watery. It's all water. Making sense? So you somehow have to make fats. You have to emulsify them. You have to make them more hydrophilic, and it is done by the formation of structures called micelles. The main function of bile salts is to emulsify fats, to solubilize them. And this is how they are doing it. We did talk, I think we did talk about bile. Yes, we did talk about bile salts. And you used bile salts in your experiment this last Monday. So I want to remind you something. Listen. Bile salts, they have two distinct bits in a molecule. One bit is hydrophilic, and then another bit is hydrophobic. Just like fats, okay? So when you have some fat molecules, okay, so these are molecules of fat. Okay, they're all hydrophobic, but bile salts stick their hydrophobic parts inside while exposing the hydrophilic parts on the outside. Okay? So inside of a micelle is a hydrophobic fatty environment. The surface is hydrophilic environment, and the surface is exposed to the watery environment of your gut. When they are solubilized, first of all, digestion of fats happens in the mouth, in the stomach, and in the intestine. Enzymes, all those enzymes carry out the same function. It's the enzyme called lipase that breaks down this fats, triacylglycerols, into monoacylglycerol and two molecules of fatty acids. Okay? These parts of triglycerides are combined, they transported into the cells of intestine where they recombine. Okay? So you kind of, like you have to, like you're moving some big item in your house, and you have to partially disassemble it and then assemble it back. Okay. Now, as you mentioned, fats are hydrophobic. What is the main chemical component of your blood? Water. And in order to transport fats in the watery blood, you need to make them hydrophilic. So how do you do that? You combine fats with proteins to form lipoproteins called chylomicrons. These chylomicrons first go into the lymphatic system, into the special lymphatic vessels called lacteals. Then they end up in the bloodstream and eventually reach cells where they are completely broken down in the process of lipolysis into glycerol and fatty acids. So what about proteins? So proteins, and I'm going to put a little note that there are also lingual lipase, the enzyme, which is in the mouth, and gastric lipase, which obviously is within the stomach. Now, what about proteins? So proteins, the digestion starts in the stomach, okay, where hydrochloric acid denatures and even breaks down some of the peptide bonds, and pepsin is the protein, the enzyme that would break down proteins further. And in the small intestine, trypsin and chymotrypsin complete the breakdown of proteins to individual amino acids. So let's take a look at this very much simplified scheme. So conceptually, though, it's fine. So in the stomach, proteins, which, remember, they're large, they're broken down into smaller bits, polypeptides. So let's say you go from 100 amino acids in the protein to several bits that will be like 15, 20 maybe, 5, whatever. And then those smaller bits, those polypeptides, are broken down into amino acids by a huge number of different enzymes. Trypsin and chymotrypsin, just 2 pancreatic enzymes, but there's way more than that. Eventually they enter bloodstream and reach the cells. Okay? Now, we're going to continue this conversation, hopefully on Monday. Okay, right now I'm going to wrap it up. We discussed how carbohydrates, proteins, and lipids are digested. Now we're going to talk about how they are further metabolized. So for the enzymatic function, often coenzymes are required, and we're going to learn the role of coenzymes such as NAD+ in the human metabolism. So first we need to discuss, and I think we talked about it before, we need to discuss the concept of oxidation and reduction. In the reactions of oxidation, the following can happen. A loss of electron, a loss of hydrogen, or an increased number of bonds to oxygen. In reduction, it's either the gain of hydrogen or electrons, or decrease in the number of bonds of oxygen. So overall, oxidation, which is characterized with the loss of electrons, loss of hydrogen, it is associated with the release of energy. You know that because when you oxidize firewood during burning, energy is released. Reduction, on the other hand, is associated with the input of energy. Since both oxidation and reduction involve a transfer of electrons, in case of oxidation, you need to move electron away. In case of reduction, you need to bring it in. The coenzyme, called NAD+, in a cell serves as a shuttle for electrons. Another name for that coenzyme is the electron carrier. So you can see on the left, it is a dinucleotide, so it has this adenine to ribose residues connected by the phosphodiester linkage, and nicotinamide. So nicotinamide can be oxidized or reduced. If it is oxidized, it loses the electrons and hydrogens and becomes NAD+. NAD+ is what's called an oxidized form. When NAD+ is reduced, in other words, it receives hydrogens and electrons, it turns into NADH, a reduced form, and this reduced form can carry electrons and hydrogens to somewhere else in the cell. So this, for example, illustrates how NAD+ serves in the cell. For example, when ethanol is oxidized by the enzyme called alkyl dehydrogenase, the electrons and the hydrogens that are taken away from ethanol effectively jump on NADH. Now, how do we know that the process that you see on your screen is oxidation? There is one bond between carbon and oxygen in a molecule of ethanol. In the molecule of acetaldehyde, there are two. So the gain of a bond to oxygen means oxidation. Now, the electrons and the hydrogens that now are accumulated in the NADH, on the NADH, can be delivered to some other chemical reaction within the cell that requires those. Another example of coenzyme is flavin adenine dinucleotide. So, dinucleotide part is this. Now, flavin part is this. And ribitol part, is this. Okay? So, same story. FAD is the oxidized form, and when it gains hydrogens and electrons, it becomes reduced form. You see two newly gained hydrogens. So, an interesting story is that riboflavin is vitamin B2, and nicotinamide is also some kind of the nicotinamide from the previous story, from this story, B3. So nicotinamide comes from vitamin B3, and riboflavin comes from vitamin B2. Okay? All good? So when FAD receives those electrons and hydrogens, turns into FADH2, the reduced form. So for the most part, FAD acts in the reactions which convert a single bond to a double bond. Technically, it's also an oxidation reaction. It specifically facilitates the reaction which converts molecule of succinate or succinic acid into the molecule of fumarate or fumaric acid. Another important coenzyme is coenzyme A. So you see this is a huge enzyme, okay? It has this active ethanethiol group, and then pantothenic acid, which comes from vitamin B5, and adenosine diphosphate. So the role of coenzyme A is to transfer various alkyl groups, for the most part, acetyl group, from one molecule to another. Okay, you can see that acetyl group binds to coenzyme, forming acetyl-CoA. And then this acetyl group can be initiated into reaction with something else. Now we're going to quickly overview the major steps, metabolic steps, that comprise the oxidation of glucose to yield ATP. So the first step of glucose oxidation is glycolysis. So glycolysis uses glucose to degrade its 6-carbon skeleton into 2 3-carbon pyruvate molecules. It occurs in the cytoplasm, and the process is anaerobic, i.e., it doesn't require oxygen. So first 5 reactions of glycolysis here, called energy investing phase. During that phase, 2 molecules of ATP are used to activate a molecule of glucose eventually, so it can be split into isomers. One of them dihydroxyacetone phosphate, and another one is glyceraldehyde 3-phosphate. So again, this is the energy investment phase because we spend 2 molecules of ATP. Now, the second phase is energy generating one, in which each of the molecules of glyceraldehyde 3-phosphate, each of these molecules is converted into molecule of pyruvate. So you can see that first of all, the product here is 2 molecules of pyruvate at the end of glycolysis. But that's not it. This reaction also generates total of 4 molecules of ATP and 2 molecules of NADH. So overall, during the energy generating phase, we get 4 ATP molecules. But during energy investment phase, we spend 2 ATP molecules. So the net yield, 2 molecules of ATP. Does that logic make sense? We invest 2, get 4 back, so the net is 2. Okay? So the overall reaction of glycolysis can be presented as you see on the bottom. You take a molecule of glucose and convert it into 2 molecules of pyruvate. During this process, we generate 2 molecules of NADH out of 2 molecules of NAD+, and we generate 2 molecules of ATP out of 2 molecules of ADP and 2 inorganic phosphates. So then the cell has to make a choice. If oxygen is available, i.e. under anaerobic conditions and aerobic conditions, cell will convert pyruvate into acetyl-CoA, this molecule. If conditions are anaerobic, pyruvate will enter the fermentation process, mostly in the skeletal muscles, and will be converted to lactate, lactic acid. So when oxygen is present, 3-carbon pyruvate is converted into 2-carbon acetyl-CoA. You see that the CoA part of the coenzyme A is now attached to acetyl through the sulfur thiol bridge. What you get out of it? You get another molecule of NADH that carries electrons and hydrogen ions. Under anaerobic conditions, bless you, so pyruvate is converted to lactate. Okay? So during this is the process of fermentation. During fermentation, the amount of ATP that's being produced is relatively small. This is why. Now, hear me out. It's not a digression. When we're talking about strenuous, high-intensity exercise. When your muscles contract, they bulge. When they bulge, they squeeze blood vessels. Are you with me? Squeezed blood vessels cannot deliver enough blood to the muscles. They become anaerobic. This is why you're getting tired so fast. If you work, if you do the high-intensity, heavy load exercises. Okay? Now, I must correct something here. No. I have studies. Lactate does not make your muscles sore or tired. It's a completely different mechanism. This is lactate? No. Lactate gives you a little bit of a burning sensation that you feel for about an hour after you got out of the gym. But then lactate is very quickly, look at this, converted to pyruvate in the liver. Okay? Now, this is also no. You breathing heavily after the exercise, not because you don't breathe heavily after the exercise long enough to repay the oxygen debt. I'm not going to get into the oxygen debt, but this is bullshit. That statement is physiological bullshit. When you stop working, like think about you doing really high intensity exercise. When you stop it and go to your car, in 30 to 40 seconds, your breathing rate goes to normal. You're not breathing hard for repaying oxygen debt. Oxygen debt repayment utilizes different mechanisms. Okay? So, following terms with the descriptions. Catabolic reactions. What do they do? Catabolism versus anabolism. Remember, catabolism is breakdown. Okay? Glycolysis. What is glycolysis? Glycolysis converts glucose to pyruvate, right? Coenzymes, substances that remove or add hydrogens and electrons. So that's going to be G, and lactate, obviously, A. So once acetyl-CoA has been produced, it enters the Krebs cycle. It is the central pathway of aerobic metabolism in glucose. Okay? So effectively, the 2-carbon acetyl group that comes from acetyl-CoA, it is oxidized, generating carbon dioxide, NADH, and FADH2. Okay? This cycle has 2 other names, tricarboxylic acid cycle and Krebs cycle, named for Hans Krebs, who discovered it. So, I always say that conceptually citric acid cycle is about counting carbons. Acetyl group has 2 carbons. An intermediate of the Krebs cycle, oxaloacetate, has 4 carbons. When they combined, the resulting citric acid has 6 carbons. And then 2 reactions remove carbons one by one in the form of CO2, resulting in succinyl-CoA, which has 4 carbons again. And then 4-carbon succinyl-CoA, after the series of steps, is converted to 4-carbon oxaloacetate, which starts the cycle once again. Let's take a look at the illustration. What I'm going to do, I'm going to write down the number of carbons in each carbon-containing molecule, okay? So we're going to start with acetyl-CoA, which has 2. Oxaloacetate has 4. They are combined, generating 6-carbon citrate. Now, citrate, you don't need to know all the steps. It's a general description. I'm going pretty fast because nobody's going to ask you individual steps. Then isocitrate, it's the isomer, also contains 6, but then isocitrate loses 1 carbon in the form of CO2, and you have 5-carbon alpha-ketoglutarate. You see, the total number of carbons is preserved. Then alpha-ketoglutarate loses yet another carbon in the form of CO2, forming 4-carbon succinyl-CoA. Succinyl-CoA is oxidized to succinate, also 4 carbons. Succinate is oxidized to fumarate, and fumarate is oxidized to malate, and malate is finally oxidized to oxaloacetate. Okay? And oxaloacetate can pick up another acetyl-CoA. during that cycle, NADH and FADH2 are produced. Remember, molecules that carry hydrogens and electrons along with a molecule of ATP. Now, so far, if you think about the whole story, for every acetyl-CoA, that enters Krebs cycle, only one ATP is generated. So 2 acetyl-CoAs that will come from pyruvate is going to be 2, but still, it's a pretty poor yield of ATP. So how do we get more ATPs? We will reveal it in a minute. So, okay, 1 turn of citric acid cycle. I don't want to go back and forth and ask you to look at the scheme. You can identify it yourself, but so far we counted 2 carbon dioxides, 3 NADH, 1 FADH2, 1 GTP or ATP, and 1 coenzyme. Now, what we finally have, I'm going to skip this slide because there's no. What we finally have, look at this. This is the summary of glycolysis, pyruvate oxidation, and Krebs cycle. Check it out. Per 1 molecule of glucose, we get 2 ATPs in glycolysis, 2 ATPs in Krebs. Not great. Agreed? Not a lot. But in terms of electron carriers, the coenzymes that carry electrons and hydrogens, we get a lot. We get a total of 10 NADH and 2 FADH2. So where are we going to use them? We're going to use them in the electron or respiratory, electron transport or respiratory chain. Those hydrogens and electrons that come from NADH and FADH2 enter the chain, getting passed from one carrier, one protein of a chain to another, release energy, which is used to indirectly synthesize ATP from ADP and phosphate, what we call an oxidative phosphorylation. Most of the ATP in the cell is produced by oxidative phosphorylation if oxygen is available. So the process of ATP synthesis happens in the cellular organelle known as mitochondria. Krebs cycle, by the way, happens there too. So mitochondria has an interesting shape. Look at this. This is schematic, okay? So this is a mitochondria. It has the folded inner membrane encapsulated in the outer membrane. Within that folded membrane are proteins of the electron transport chain. Okay? And there are 2 more structural elements, coenzyme Q and cytochrome C, that are not attached to the membrane but rather float in it, which allows them to shuttle the electrons between protein complexes in that chain, because these complexes are anchored to the membrane. So this illustration shows you what happens at the electron transport chain. Let's break it down. So first, NADH loses its electrons and hydrogens. So it is oxidized, and the electron enters the first complex. At the second complex, FADH2 is also oxidized. Losing its electrons and hydrogens to the second complex. And then these electrons, look, they are transferring from one complex to another, just like a ball. Imagine a bowling ball that you push off of the top of the staircase. Am I making sense? And they just go step by step. Okay? At the end of this chain is the oxygen molecule, also known as the final electron acceptor. Okay? Good? Final electron acceptor. Now, why do I tell you this? All these transfers of electrons from one complex to another release energy that is used for the pumping of hydrogens across the membrane. So now we have high hydrogen concentration on one side of the membrane. Does that make sense? And low hydrogen concentration on the other. Okay? Now, you may remember that when we have this distribution of concentrations, it's called the gradient. Okay? So here's your hydrogen on one side in the intermembrane space. It will diffuse right here through the enzyme called ATP synthase. It will diffuse through that enzyme into the matrix of the mitochondria. So this overall process is referred to as chemiosmosis. Each complex pumps protons using energy that was derived from the oxidation of NADH and FADH2. The electrochemical gradient that was created, it's called electrochemical because it's a gradient of charges, and hydrogen is a chemical, okay? It facilitates the diffusion of hydrogen ions to the matrix by passing through ATP synthase. So the energy of that diffusion provides energy required for ATP synthesis. So effectively, when hydrogen ions diffuse down the gradient, that releases energy. This energy that is used for the ATP synthesis. So NADH gives its electrons and hydrogens to the very first complex in the chain, so it produces, each molecule of NADH gives energy enough to produce 2.5 ATPs. FADH2 gives its electrons to the second protein complex in the chain. So it gives enough energy to produce just 1.5 ATPs. Now, considering the fact that glycolysis, which by the way occurs in the cytoplasm, produces 2 NADH, okay, and 2 ATP molecules. This is the calculation of how much ATP is generated from glycolysis during that oxidative aerobic process. So 2 NADH molecules give you 5 ATPs, because each NADH, does that make sense? Because 1 NADH gives you 2.5 ATPs. Okay? And then 2 ATPs are also generated by the direct phosphorylation. So overall, this is your summary of glycolysis. Glucose gives you 2 pyruvates and 7 ATPs. So 2 pyruvates then are oxidized to acetyl-CoA, generating 2 NADH. Since every NADH produces 2.5 ATPs, you get 5. Now, citric acid cycle gives you 3 NADH, 1 FADH2, and 1 GTP, which is kind of ATP, per turn. But each molecule of glucose generates 2 acetyl-CoA, which means there are going to be 2 turns of the citric acid cycle. So 3 NADH in 1 turn, 7.5 ATP, 1 FADH2 in 1 turn, 1.5 ATP, and then another ATP from phosphorylation. So you get 10 ATPs per 1 turn of the cycle. Does that make sense to you? 2 turns give you 20 ATPs. So now, when you put it all together, glycolysis 7, oxidation decarboxylation 5, and citric acid cycle 20, you get 32 ATPs. This is the amount of ATP that can be produced from 1 molecule of glucose in this aerobic respiration. Let me see. I gather that we're going to leave oxidation of fatty acids for our last reasonable class on Monday. Okay? Does that make sense? I will check, actually, maybe I will record it and post so you have more time to get ready for the exam. Sounds good? Okay, folks. When we met last time, we discussed the whole breakdown of glucose, the oxidation of glucose, right? So, glucose is the most versatile energy molecule because it can be fermented when there is no oxygen, can be oxidized when there is oxygen. Fats can only be broken down in the presence of oxygen, effectively. So they go through so-called beta-oxidation. And our goal is to figure out how conceptually it works. So effectively, just a quick reminder, folks, when you have triglycerides or triacylglycerols, they are, during the process of lipolysis, broken down to glycerol and fatty acids. So glycerol is converted into glyceraldehyde 3-phosphate, which is one of the intermediates in the process of glycolysis, and fatty acids undergo beta-oxidation. So why do we call it beta-oxidation? Look, the fact that they are fatty acids means that there is a carboxylic group at one of the ends. Do you follow? The carbon atom that is closest to that carboxylic group is called alpha carbon. The next is beta carbon. So what effectively happens, check this out. This chunk, these two carbons, are broken off, and the carboxylic group appears at the beta carbon. We are not going to go through all these steps mechanistically. Here's what I want to highlight. So fatty acids are hydrophobic. Does that statement make sense to you? You know, they're fatty, right? So in order for them to be transported within the cell, they have to be made hydrophilic, and they have to be chemically activated in order for being broken down. So coenzyme A that we already learned about, pretty multifunctional molecule, combines with a fatty acid, okay, and you get fatty acid coenzyme A here, okay. Then in the mitochondrial matrix, fatty acids go through beta oxidation. It is a cycle. There are 4 reactions that convert beta carbon, okay, to a keto carbon. Can you see that? And at the same time, acetyl-CoA is formed. So for this, you need an extra coenzyme A, and you need NAD plus and FAD in order to collect electrons taken out during this process, during oxidation. Make sense? So one more time. Fatty acids being oxidized. Electrons and hydrogens are picked up by NAD plus and FAD. During beta oxidation, acetyl-CoA is formed, and the chain in the fatty acid is shortened by 2 carbons. So you gnaw off 2 carbons at a time. Does it make sense? Take a look at this. So this is your first molecule. It has 8 carbons, right? You chip off 2. You end up with 6, then 4, then 2. And at each step, you generate acetyl-CoA, which can then enter the Krebs cycle and be further oxidized. So all in all, beta-oxidation per molecule gives you more energy. It's not a fair comparison though, because we need to compare per carbon rather than per molecule. So look at this. Let's talk about stearic acid. Okay? So what you're going to get is 9 acetyl-CoAs in 8 oxidation cycles. Stearic acid has 3 times as many carbons as glucose. So now take a look at the energy yield. One cycle, you generate 1 NADH, so it's 2.5 ATPs. FADH2, 1.5 ATPs. And acetyl-CoA, which during the oxidation in the Krebs cycle generates 10, which is quite a lot. So if you look at capric acid, okay, which contains 10 carbons, you have 5 acetyl-CoAs. Each acetyl-CoA brings you 10 ATPs, so 50 ATPs. 4 NADH and 4 FADH2, which gives you extra 10 and 6 ATP respectively, so total of 64 ATP. Okay? So far, do you follow? Now, here's the thing. I want to do these quick calculations for stearic acid. So stearic acid has the same number of carbons as a single glucose. Okay? So, if you think about the process of beta-oxidation, chopping off 2 carbons at a time, the total number of acetyl-CoAs will be 9 for stearic acid. Does that make sense? You divide 18 by 2, 2 carbons in each fragment. Do you understand what I'm saying? Yes? So 9 acetyl-CoA. How many cycles? 8. Okay? So that means 90 ATPs from acetyl-CoA and 32 ATPs from 8 cycles. Each cycle brings you 4 via NADH and FADH2. Minus 2 ATPs for activation. Do you follow me for now? I'm just giving you an example. I'm not asking you to calculate that stuff on the exam. As long as you know what beta-oxidation is, you're good. I don't expect you memorizing how many NADH generated, how many FADH2 are generated, and so on and so forth. But conceptually, I want you to understand that it's breaking down a fatty acid to acetyl-CoA. So, the resulting number, 120 ATP. 1 glucose gives you 32 max. So, 6 carbons of glucose give you 32. 18 carbons of stearic acid gives you 120, which is 25 more percent per carbon, if you do the math. Fat is better source of energy than glucose is. So unsaturated fatty acids, no FADH2 formed, slightly less energy. Okay, does that make sense, everybody? Again, I'm asking about conceptual learning. I promise you this, you can seriously take me up on my word, and it's going to be recorded, and I'm not editing this shit out. If there is a question in which suddenly you need to count how many ATPs will be produced from a molecule of myristic acid, this question's out. Okay, if it is on the exam, I forgot to take it out or whatever, this question's out. So ketone bodies, it's a very interesting idea. So let's talk physiology for a little bit, just a little bit. Okay? Okay. Let's talk about this. I'm actually going to go all the way back. You don't have to, but it will be easier for me to have not this. Okay, never mind. This. Okay, I don't like this image, but I have to work with whatever I have. So listen. Imagine that you decided to go completely carbohydrate free. You know, your diet is salad, celery, avocados, and chicken breast. Does it make sense so far, what I'm trying to tell you? So, do you get any glucose with the diet if you go completely carb free? Do you get any glucose? No. Can cells in your body exist without glucose? Most of them can, actually. They can use fat. There is a subset. There's one organ that does not do well without glucose. Any guesses which one it is? Organ that basically demands glucose for its function. Do you have munchies when you study? No, seriously, do you have munchies? Yeah? Well, that too, but... And you're not munching on avocados or chicken breast. You're munching on cookies and chocolate because your brain cannot be active without glucose. Does it make sense to you? So you go completely carb free. All cells can work on something else except for the brain. So what do? Your cells, your liver and your kidneys start making glucose from scratch. Clear? The resource for this is oxaloacetate. So you start using oxaloacetate to make glucose. Are you with me? If you take out one element of the cycle, can it continue? No. You don't have the intermediate, right? So what you end up with, and remember, you are eating a buttload of fats. So you have no deficiency of fatty acids. Are we clear so far? Yeah? The breakdown of the fatty acids results in the formation of acetyl-CoA. Otherwise, this molecule would go into the Krebs cycle, right? But your Krebs cycle isn't functional because you took one element to make glucose. So somehow you need to oxidize acetyl-CoA. So far, am I making sense? So this happens in the process of generating ketone bodies during the process known as ketogenesis. Okay? So what do you form? You form acetoacetate here, beta-hydroxybutyrate, and acetone. Am I making sense so far? Yes? The main molecule that is made is acetoacetate and hydroxybutyrate. When do they form? As we mentioned, you accumulate large amounts of acetyl-CoA. Okay? And what do you do with these bodies? Well, there are two options for for getting into ketosis. Option one, not healthy. When somebody has diabetes, which is poorly controlled, am I making sense? When people have diabetes, they can't get the glucose out of the blood and in the tissues. Tissues have to use fats. And when they're running exclusively on fats, they develop ketosis. Have you heard the term diabetic ketoacidosis? People who have poorly controlled blood glucose often have the smell out of the mouth that smells like what? Acetone. Does that make sense? It smells like acetone. Fruity smell. Okay? You can detect ketosis by doing the test strips on the urine. Okay? Is all second option for ketosis. Have you heard about keto or Atkins diet? No? Yes? Yeah. Yeah, keto diet. You exclude carbs. Period. You can eat as much fat and as much protein as you want. Okay? Drawbacks. Unknown yet. Pluses. When you are in ketosis, in dietary ketosis, first of all, it's not the same as diabetic ketoacidosis. It's not going to shut down your brain. Make sense? If you move around, if you do all of this, like if you are physically active and you are in ketosis, the huge benefit, you're going to be losing weight, and not just any weight, you're going to be losing fat. Does that make sense? Health drawbacks. Unknown yet. I'm not saying there's none, but so far didn't appear to have any. What sucks about ketosis? Getting into it is a pain. You feel like crap. You constantly feel tired. If you stick to it though, you become independent of carbs and your aerobic endurance is insane. And you become very lean. Am I making sense? Are you going to pass ketone bodies in the urine? Yes, you can. You will. So you basically I know some maniacs that put themselves in ketosis and check the ketone levels in the urine. Okay? I tried it once. It's horrible. I can tell you this. If you're miserable, if you feel tired, again, you feel tired, your actual endurance and your strength, they do not change. Just you overall feel tired. And if one tiny piece of chocolate feels like cocaine in terms of enjoyment, you're in ketosis. Okay? Yeah, obesity. Okay, folks, the reason for obesity in the United States, there are several factors. I'm just going to tell you real quick. Poverty and stress. Cortisol levels increase appetite, and people also want palatable food because they stress. Nobody wants to eat kale when they stress. So they eat highly palatable food, which happens to be mostly carbs, mostly simple carbs. Okay, poverty. Not enough time to cook. Not enough time to select much, prepared meals are much easier, and they contain a lot of carbohydrates because it makes them really tasty. Does that make sense? If you're curious, right now, we broke through 30%. More than 33% of this population are obese, which classifies as the BMI over 30, body mass index. Okay? And another third of US population are overweight. So only a third of the US population have normal body weight in the range between 20 and 25 BMI. I must say this. Hear me out. What is the state with the lowest levels of obesity? I'm going to do some quiz here. You can that is guessable. Yes, it's Colorado. Okay? Because it's expensive, so much less poverty, and usually people who move there like to move around. Okay, like to walk. What is the state with the highest level of obesity? Arkansas? It's up there. It's a competition. It usually it's like a unholy five. West Virginia, Louisiana, Mississippi, Arkansas, with Alabama being very close behind. Okay, if you look at the distribution of obesity in the United States, there is what I call an obesity belt that starts around at Texas and goes all the way down to Florida and up there in Georgia's there, South Carolina. Basically, south of the Mason-Dixon line, things do not look good. Okay? Genetics, poverty, genetics, people with African American background, ethnic background, people with I don't want to say Latino, specifically Native American background. So when they say Latino, it depends. If you like mostly Spanish, much less risk. If you're mostly Native American, much more. And specifically Pacific Islanders, Samoa, they have predisposition for obesity. Okay? Yeah. Watch what you eat and move around. Leptin, my favorite hormone. I'm going to say a few words about it and let you go. I always joke that leptin is the only hormone which effects can be voluntarily overruled. Let me explain to you. When you are stressed, you produce cortisol. That's it. You cannot do anything about cortisol effects on your body. Something like that. So leptin is produced by your fat tissue after the meal. And basically it's a signal to put down the fork. Does that make sense? Now, if you imagine you are at a party. Oh, come on. Thanksgiving. The moment when you received the signal, put down the fork, you're full. The moment you did put down the fork, I bet there were several hours. At least that was what it was for me. Okay, so we can overcome that signal. Make sense? And one of the reasons why there's a nutritional advice when you get off, away from the dining table, you should be a little hungry, because it takes time for the leptin to kick in. It takes time for the fat cells to get the message from the liver, and it takes time for them to produce leptin, and it takes time for leptin to signal to your hypothalamus that you're full. Okay? Yeah, and leptin also regulates, to some extent, the metabolism of leptin increases the breakdown of fat because there's too much fat coming in. Couple of notes. How do you study obesity in mice? One way is to feed them, but if you want it pure, there are mice that do not have the gene responsible for leptin production. Does anyone know an average weight of a mouse, like a regular garden variety lab mouse? What do you think? No, that's a really fat rat. It's more than a pound. No, an average mouse is like 2/3 of an ounce. It's about an ounce. Okay? An average adult lab mouse. Mice who have leptin deficiency can weigh 3 times that, which is insane. Like if you think about like an average male going from 180 pounds to 540, that's a lot of pounds. Those mice they have a form of a circle. They have troubles walking because they're so fat. Okay, it's a pretty sad story. But yeah, leptin is the signal for you to put down the fork. Okay.